Good morning, everybody, and thank you, Josh. Thank you to the Alliance for Regenerative Medicine and Maury and Piper and Cowan. It's a pleasure to be here today and introduce Homology Medicines to you. This is really the core tenets of the company, what underpins what we're doing here. Homology's approach, we think, is a highly differentiated approach to both gene transfer and gene correction or gene editing. This is based on a newly discovered class of AAVs that was discovered at the City of Hope, isolated from human hematopoietic stem cells. This is a single nuclease-free AAV platform that can do either in vivo gene editing or gene correction or gene transfer. The focus of the company is really based on speed to the clinic, initially in monogenic rare disorders, initially through gene transfer. And what this technology can do through AAVs that we have IP on is to mediate gene correction through a naturally occurring DNA repair process known as homologous recombination to ultimately cure disease. We have core intellectual property from the City of Hope, exclusively licensed, and an expanding IP estate. The team we've built are mainly former Shire execs with gene therapy experience, importantly, in bringing novel discoveries from the bench all the way through to commercialization. And I think that's very important to combine this technology with an experienced team. So these are the AAVs. We call them AAV HSCs for hematopoietic stem cells. Right now we have a panel of 15 of them, and they belong to Clade F. We have U.S. Issued composition of matter patents on these AAV vectors. And this is really a panel of vectors that we can select from to do either gene transfer or gene editing. And really the whole idea here is because we really have a lot of experience in the development of rare genetic disease therapies, is to use the flexibility of this platform and understand the disease biology and then pick whether we want to do gene transfer for a particular disease or whether we want to do gene editing or gene correction. So on the gene editing side, we have flexibility in that we can do gene correction with entire genes or single nucleotide corrections, or even knock out certain genes in autosomal dominant diseases. On the right-hand side, gene therapy, we can use these AAVs, which have superior biodistribution, can cross the blood-brain barrier very well to really deliver genes that will express the proteins that are needed in that tissue. Really, we see this technology as immediately developable. And what do I mean by that? I mean, it can be brought to the clinic fairly rapidly because it is a single platform technology. There's a single AAV that carries the payload. So there's not a multi-component system here. Because it's homologous recombination, there's exquisite precision in where you want to make your correction in the genome. And that's very important here because one of the biggest issues here in gene editing is, are you doing what you want to do, and are you not doing something bad to the genome somewhere else? And that really is important in terms of making sure you know what your off-target profile looks like. We are seeing very good in vivo efficiencies of gene correction, 10% and above, which gives us the ability to go after diseases with an in vivo approach versus an ex vivo approach. And from a manufacturing point of view, this is fairly straightforward because it's just a single AAV that you're manufacturing, not a multi-component system that you have to put together at the end. So this makes manufacturing and ultimately commercialization very viable. This is our platform. There's 3 general areas we're working in. At the top, you can see inborn areas of metabolism, so liver diseases. Initially, we're going to start out with PKU and gene transfer, ultimately moving to a gene editing platform there. Because these AAVs are isolated from human hematopoietic stem cells, or CD34 cells, it's kind of a natural to go after the hemoglobinopathies. And we can do this with an in vivo correction approach versus ex vivo, which is going on right now. And then finally, because of the properties of these AAVs to cross a blood-brain barrier, and because we've also licensed technology from Caltech, from the lab of Ben Deverman, that improves the ability to cross the blood-brain barrier, we feel like monogenic rare CNS is an area that we can really help. And as a team from Shire, we have a lot of experience in developing monogenic rare CNS drugs. So we've launched really a year ago in April, and this is a 5 a.m. back company, which is right across the street, as well as an Arch back company, Deerfield and Temasek. We've built our team. We're about 32 folks right now. We've got a fully staffed R&D team. We have our own labs. We're out in Bedford, Mass. We've got our indications selected. We have a development candidate already for our lead program, and we're already in IND-enabling studies. We already are working hard at gene editing, gene correction using these vectors and have proof of concept there. And we continue to build out this platform and discover more of these AAV HSCs. And as I said, from an IP perspective, we have a single founder at City of Hope with a single IP estate that continues to get stronger. So we feel like we're in a good position to really help patients with rare genetic disorders, which is a passion for me personally and for our team and for our company. So thank you. I'll have a Q&A with Josh now.
Excellent. Well, thank you. So maybe the first question as we think about various AAV strains and subtypes, I think we generally associate them with different capsids. Have you found a strain that has more efficient homologous recombination, and if so, what is that attributed
Right.
to?
So what we know is that this family of clade F AAVs has this ability to do gene correction or gene editing by homologous recombination, and really this is not something new. This has been known since David Russell published a paper in 1998 that you can mediate HR by AAVs, but that efficiency is pretty low. It's 0.5, 1%. And so with these AAVs, and this was the discovery that was made by Dr. Chatterjee at City of Hope, you can really see efficiencies that are upwards of 10, 15, and north of that %. So that gives you the ability to do in vivo gene editing. And importantly, you can do this with the endogenous promoter. You don't need to go into a safe harbor locus in order to go after some of these diseases.
Do we know what aspect of the plasmid itself is driving the improvement in HR?
Right. So what we think is going on here is a structural capsid amino acid combination that's leading to the binding to a receptor on a cell that's signaling for increased homologous recombination. So that's the hypothesis right now, and we're working hard on elucidating that, but it's pretty clear that that's associated with these capsids from this clade, and it's pretty clear that you require these homology arms, which flanks the corrected sequence that's in the payload that's driving this process.
And what do you see as the key challenges as you move from kind of bench to bedside, and how are you kind of gleaning as much knowledge as you can from the experiences that have gone before you to make sure that translation is efficient?
Sure. No, I think one of the things I think that we bring to the table is, first, that experience in rare drug disease development. There's a lot of things that I think that are underappreciated that we know in terms of recruitment of patients, identification of patients, working with patient advocacy groups. So those things, I think, are a challenge that I think we're prepared for. I think from the science side, it's really important for us to get our ducks in a row in terms of manufacturing. That's going to be a theme today. You're going to hear from a number of companies to making sure, very early on that you have that squared away and you're able to have a reproducible process that you can deliver to patients in a reproducible way.
On that topic, I know a number of companies have gone down the route of building their own manufacturing capacity. I guess it seems like a lot of reinventing of wheels. Is that the path you think ultimately everyone's going to have to take, or are there going to be more efficient, rapid paths forward as we learn more about manufacturing?
Yeah, I mean, I can tell you from my experience, manufacturing in any field is difficult. We know the challenges in the enzyme replacement field that have been experienced. But I would say initially we're going to have to rely on CMOs because it's pretty nearly impossible for a new company to build its own manufacturing facility. So you're going to have to learn to select CMOs that are going to be able to deliver for you. And I think the whole science of manufacturing in this field is absolutely increasing every year. So I think initially you're going to have to do that. Then it becomes a choice for the company. Is that best for the company? What's the indication you're going after? What's the scale you need? So all those things I think you have to take into account before you make that kind of decision because it's a large capital expense. And if you're going to your Series A or Series B investors and saying, I want to build a building over here to manufacture something, they're going to look at you and say, really? So, you need to kind of demonstrate that that's absolutely required, and you need to have the capital to be able to do that.
And then as you look at your unique capabilities, what was the process, and how do you continue the process of matching that to your target indications? What were the criteria that you used that led to your
when the management team came on board in April, they had a list of indications, and we kind of said, no, we're not going to do those. We just had a lot of experience and probably had 2,000 meetings on what indications you should go after and with what technologies. Some of the folks in the audience are laughing because they've been in some of those meetings. So we had that kind of domain expertise to know that we should go after a disease where a small amount of expression is going to give you a normalization of a phenotype. So you're going to see a cure with maybe 5% expression or 8% expression.
I will just stop and see if there are any questions. Please.
Absolutely, yeah. They're isolated from human CD34 cells. Right, so they're tropic for mouse cells as well as non-human primates. So those are things that we've done, experiments we've done to show that they do have tropism for mouse, human, monkey.
So they're not just human specific.
Right, that's correct.
Maybe for those who aren't as familiar with homologous recombination, give us a quick overview of how the technology actually works and how you can gene edit without a nucleus.
Right, so homologous recombination is going on in everybody in this room right now, in all of our cells. It's a DNA repair mechanism, and it's basically the ability to swap similar strands of DNA from chromosome to chromosome. But you can engineer homology arms that flank a corrected sequence, and that's our payload, and target very specifically a mutated gene. And so if those homology arms to the left and to the right of that corrected gene are perfectly matching or complementary for the flanking sequences of the mutated gene, if you bring those strands in close proximity, you'll have strand exchange. And so that's the way that you design the vectors such that they can be highly specific, highly precise, with very little to no off target.
And this can be done both in vivo and ex vivo.
That's right. So we have data showing very clearly in vitro and very clearly in vivo that you can mediate either single nucleotide or whole gene insertion into the genome for correction.
As you evolve the pipeline, one of the big unknowns is how the business model and the commercial model will work. How do you factor that into your planning? And as we build models, there's still a lot of question marks in it.
Sure. So we're very fortunate to have a commercial rare disease expert as our COO, Sam Rosti from Shire. So already he's building in the thinking around the indications we're selecting. Are there enough patients there? Are we going, I think the key in this panel this afternoon on this, the key is, are you going to be able to deliver a transformational cure or a long-term treatment that's going to make a huge difference in the lives of patients? That ultimately is going to be the decider of whether or not you're going to have a commercializable product or not. That's really kind of, I think, the common denominator when you're trying to assess whether or not you're going to see success in the clinic and success commercially.
I guess, and the way you describe the platform, it sounds like the capsid itself is an important part of this, and so is there an opportunity to move away from AAV into something that can be re-administered, or AAV is really part of this package?
I think that's always a possibility. Once you figure out what the capsid sequence is that's driving this, you can come forward with a synthetic AAV, if you will. I think that's down the road. What I like about this platform is that it's natural. These are naturally occurring AAVs. This is a naturally occurring repair mechanism. And in my experience in drug development, the more natural you are and you can harness that, the better chance you have for success in the clinic and commercially.
What's the limitation for size?
Standard AAV, so 5 kb.
You don't need any of the genome in order to maintain the editing?
Yeah, so you can, we can fit about 85% of the genomes into these capsids. So no different from AAV2, 8, 6.
To what extent does that limit the selection of diseases that you look at?
I'm sorry, say
To what extent does capacity limit the selection of diseases?
So I said, we've done a genome scan, and as I said, 85% of all the genes in the genome fit into this family of AAVs. So we have a pretty wide spectrum there. And then even with some of these larger genes like dystrophin, there's strategies we can use to go after part of that gene versus having to put in a whole gene.
And last question, do you have to replace the whole gene, or based on this approach, can it just correct a short piece of the gene and leave everything else intact?
You don't have to correct the whole gene. We can in some diseases, like the hemoglobinopathies, but in many cases you can go in and make a small correction into the gene where there's, say, a common mutation like delta F508 and CF.
Great. Well, excellent. Thank you very much. Looking forward to
Thank you,
following the
Josh.
progress.